Now I will show you the molecular mechanism of this class switching. A group of scientists led by Professor Takasu Honjo, very eminent immunologist, working on a cell line, and they found that a particular B cell line form do not perform or show class switching or isotype switching. And very unexpectedly they also found that those B cell line do not perform this class switching or isotype switching. They also do not perform somatic hypermutation, another another event of B cells by which actually the affinity of the antibody increases. So they found that there is a mutation in a protein that they call AID, that means activation induced cytidine deaminase. This enzyme is very important for these two events, that is class switching or isotype switching and the somatic hypermutation. This is the heart of the two mechanisms. And this enzyme is actually activated by a binding that is called CD40 ligand and CD40 interactions. When these two molecules interact, the AID, activation induced cytidine deaminase enzyme, actually activated. So that this name is activation, because during the activation of B cells, the CD40L and CD40 interact. I have already discussed about in my B cell activation class, CD40L from the T helper cell and the CD40 from the B cells. When these two molecules interact, they activate this enzyme, and this enzyme actually perform its function. This is the molecular mechanism behind this class switching. So let me draw the constant heavy region. So I have already discussed about that. The class switching occurred between this constant heavy domain of the antibody. So let's assume this is a double-stranded DNA, and here is the segment, gene segment for constant heavy domain for mu. That means from this DNA segment IgM antibody will be produced, and there is another gene segment, I am making it in black color, that is constant heavy for delta region. That means from this segment IgD antibody will be produced. And as I have made IgG in red color, so I am making the gene segment also in red color. Let's assume from this gene segment, this is a C constant heavy for the gamma. Here from this IgG antibody will be produced, and IgE antibody will be produced from this segment. So it is constant heavy for epsilon, that means IgE antibody will produce from this segment, that means constant heavy region. And this is the constant heavy for the alpha, that means IgA antibody. And this is the heavy chain, this is the heavy chain gene segment, okay. And this segment, there are 5 constant heavy region are present. And as I have already mentioned that when this B cell actually develop, or they actually go through VDJ recombinations, so they have already performed a process by which they have make it variable region, that is the VDJ. So that V, D and J, that is already present in this heavy chain, that actually form the variable heavy. This region will not change, but this region can be switched. So this is called class switching. The scientist Takasu Honjo, they found that there is some region present in this gene segment that called switch region. They found some DNA segments approximately 20 to 80 base pair, and this switch region is actually G-rich sequence, and they call this switch region into two. One is called donor switch region and another they call acceptor switch region. These are the DNA segments. They found that those switch region present 5 prime before this IgM and IgD 5 prime region of the DNA segment. They called it donor switch region, and there is no switch region between this M and D. There is no switch region between this constant heavy mu and constant heavy delta, because they actually inherited as a one. Okay, in mature B cells I have already shown you that this IgM and IgD antibody are already actually expressed togetherly. So there is no switch region present between this IgM and IgD constant heavy segment. Then there is another segment of this gene, that is switch region, present before this IgG, before this IgA and before this IgE. This is the switch region. This is the donor and this is the acceptor region, acceptor switch region. What happens when these B cells get activated by Th1? The Th1 secrete IFN gamma, and that actually switches the antibody from the IgM IgD to IgG. If it is Th2 cells, then it will switch into Ig. So depending on the type of interleukin or chemical secreted by the T helper cell, it can switch its antibody. So let's assume Th1 cells is activating these B cells, Th1. So Th1 will secrete IFN gamma. So this gene actually this gene segment, now these B cells actually produce IgG type of antibody. How this process occur? The main event of this class switching is to make double strand DNA break. So what I have to perform here, a double strand break should be, and in this region there should be a double strand break. Let's assume if this B cell will produce IgA antibody, then the double strand break will occur here, and if the IgE antibodies require, then double strand break will occur here. As I am mentioning here that IgG will produce, so I will not show here the double strand break. So double strand break will occur, and in this region, this intermediate region will actually form a circular domain. So this here will be a double strand break, here will be a double strand break, and the DNA segment will become like this. Let's assume this is the DNA intermediate DNA segments, and this donor half segment, this is the donor half segment, and the acceptor half segment will actually bind with each other, and the two constant hemi domain of the DNA, like this is the C constant of mu and this is the C constant heavy domain of the delta, will present in the intermediate segment, and the rest of this DNA segment will bind each other. How this occurred I will show you. So at first, when this interaction CD40L and CD40 interact, this AID enzyme gets activated, and what happens when this B cells actually produce or transcribe antibody, this double strand segment of DNA becomes open. So let's assume this is the template strand and this is the non-template strand. Okay, and this region is the switch region. Here the donor region. Let's assume this is the donor switch region. Donor switch region. As I have mentioned that donor switch region is GG sequence. So here in this template region there will be G sequence. So in this non-template region there will be C sequence, many C sequence. So let's assume this is A, C, C, T, C, G, C, C, T. Okay, let's assume, and here the G sequence are present. Alternative or complementary DNA sequence are present. So in this non-template strand, from this template strand RNA will produce. So AID actually react non-template strand, and what happens when cytidine nucleotides are reacted by AID enzyme, the cytidine actually become uridine. This enzyme actually deaminates, remove a amino group from the cytidine nucleotide, and it becomes uridine. So when this AID will react on this donor region, what happens? This C nucleotide, all the cytidine nucleotide will become U. All the C cytidine will become U. Now, in our cell, there are enzyme called uridine N-glycosylase. This enzyme can sense that if there is any uridine present in the DNA, that means there is some problem, because uridine can present in RNA, because RNA consists of 4 nucleotides, that is UGC, and in the DNA there is ATGC, no uridine is present. But if this enzyme can sense that there is uridine present in the DNA, it actually sense that there is problem, and this enzyme actually remove the uridine, so it will remove. So when this enzyme react on this strand, they remove the uridine. Now this situation, when there is no base present in the non-template strand, this situation is called abasic site, and this abasic site is also a alarm that our cells can sense that there is a problem, because no DNA should be abasic conditions, because abasic means there is some problem. So there is already another enzyme is actually dedicated to sense this type of situations called APE1 endonuclease. So you can see in this class switching mechanism, three enzymes are responsible. One is AID, second is urine glycosylate, and the third is APE endonuclease. This APE1 endonuclease can sense where is the basic site, and it will cut in that region where there is no base, and eventually this APE endonuclease actually cut in the template strand also. Eventually it will cut the template strand. So ultimately, after this mechanism, what happens? Double strand break will occur, and this is occurring in the donor switch region. This same mechanism, the action of AID, urine and glycosylation, and AP1 endonuclease also occur in this region. So what happens? Here will be a double strand break, here also will be a double strand break, and this intermediate sequence will actually join with each other, and the rest of the DNA segment I am showing you. This is the VDJ sequence, V, D, and this is the J that actually make the variable heavy region actually will remain. And now there is no IgM and IgD DNA segment, but at first there will be a IgG or constant heavy for gamma. Now I am showing here this segment will join, and there will be this region also present. This antibody constant region could be present. See, constant heavy epsilon and constant heavy for alpha. Now you can see this B cell, so there is another segment is produced, because the half D segment and the half acceptor segment will be present here. This is the D and A joining. Now this will be the donor switch region in these B cells. When the B cell go through the first class switching, this half donor sequence and the half acceptor sequence will join, and this will form the donor sequence region another time. And here is already present the acceptor switch region. So now you can understand these B cells. Once it actually switched into IgG, it can switch into IgA, IgE if it requires. Okay, so if there is any class switching and if this B cell is producing IgE type of antibody, then break will occur. This double strand break will occur in this region and in this region. Now this IgG or C, constant heavy gamma and constant heavy alpha region will actually form look like this, and these two regions will join each other.